Hello everyone, welcome back. So this is week 5 of the NPTEL course Immunology and I am Bias Mukherjee. I will be talking about antigen recognition, processing and presentation in today's video. Okay, and this course is coordinated by Professor Ghosh and Professor Ganguly from the Department of Bioscience and Biotechnology. And you know that we have been meeting for the past 4 weeks. We have talked about antibodies, antigens, we have talked about immune responses. Okay, and in today's class, we are going to talk about antigen recognition, processing and presentation by MHC molecules in details, okay, in a lot of details. So in last class, we had started with MHC molecules and we have talked about the structure and the genetic rearrangement that gives MHC its variability. Okay, today we are going to talk about the functions of MHC and how it happens. Let's start with today's class, okay? Right. So if you remember, MHC molecules are of two types. There is class 1 MHC and there is class 2 MHC. And the purpose of MHC molecule is to present the peptides that are antigenic peptides that have been uh, found in the pathogens that have entered into our body, and this pathogen was engulfed by, let's say, a macrophage or dendritic cell, or it can be from a virus infected cell, and that virus infected cell is producing these viral proteins. Okay, now there are two types of MHC molecules, and if it is a class 2 MHC molecule, it is going to present the peptides that are exogenous. So they have been brought within by this pathogen from outside. If it is a MHC class 1 molecule, it is going to present the peptides that are synthesized inside the cell by the viruses, or it can be any tumor antigens that are made by tumor cell. Okay. Now we will look at MHC class 1 and class 2 once again. So this slide was there in your previous week also. So if you remember, this MHC class 1 and class 2, they are structurally a little different. So MHC class 1 is made up of two chains, alpha 1, alpha 2 and alpha 3 make up one, and then beta 2 microglobulin is the other one. And then there is this peptide binding cleft. So there was very significant distinction in the two peptide binding clefts of the two MHC molecules. Okay, and MHC class 2 has alpha chain and beta chain, and then we are going to talk about more of this starting in today's class. Okay, now the chain composition. So basically MHC class 1 is polymorphic alpha chain. So that means the alpha has three variants, alpha 1, alpha 2, alpha 3, that we saw in the figure, and there is an invariant beta 2 microglobulin that we saw, right? Then you have this MHC class 2, which is polymorphic, that is alpha and beta. These are there, alpha 1, alpha 2 and beta 1, beta 2. Peptide binding groove is also different. For MHC class 1 molecule, it is closed at both the ends, and the hydrophobic contacts between the peptide molecules and the peptide binding cleft is very, very important here. Okay, plus you have the class 2, the peptide binding cleft is more or less open ended. Okay, and that is why it can fit in longer amino acid stretches. So around 13 to 20 amino acid peptides can be fitted in your second MHC class 2 type. Okay, and for the MHC class 1 type, 8 to 10 amino acid stretch can fit in at a time. And then the cell distribution is MHC class 1 is there in all nucleated cell. MHC class 2 is specifically there in antigen processing cell or APCs, right? Now you have the source of the peptide. This I am sure you remember already. So for class 1 it is endogenous, for class 2 it is exogenous peptide. Okay, and T cell subset activated. So class 1 activates cytotoxic T lymphocytes with CD8 receptor and your co-receptor, and your class 2 MHC, this activates helper T cell with CD4 plus co-receptor. And then the human genes and all this we are going to talk about in today's class. Now see, this MHC complex, this is present in chromosome number 6 in humans, and it is also known as HLA. So what is HLA? HLA means human leukocyte antigen. So MHC and HLA refer to the same thing, okay? This stretch of genes that are present in the same locus. So if you remember, locus is a region in the chromosome, a stretch of, it's basically a position in the chromosome, okay? There a lot of gene segments are present together, clustered together. That is your locus. Now MHC has three classes of genes. The first one is, so there is class 1 genes, which are called HLA A, B and C. Then there is class 2 genes, which is called HLA DP, DQ and DR. Now what is this D? Please read about that, okay? Then you have, so these again have two separate genes each, alpha gene and beta gene. We will see this in the figure in the upcoming slide, okay? Now, these sequence genomes reveal hundreds of alleles per locus. So basically, there are a lot of sequence variations within these genes in the population. Okay, so your, let's say this DP region of the gene in your body will be different from that in my body. Okay, this is how throughout the population there are multiple alleles of these genes. Okay, and that gives MHC its diversity. Now MHC is also very diverse, just like your B cell and T cell diversity, receptor diversity. MHC is also very, very diverse. Okay, we'll talk about that in today's class. Okay, for example, hundreds of DPB1, dozens of DPA1. So basically this DP, this also has variants, A1, B1, etc. All of these variants are there. Okay. Now each individual carries only a subset of known alleles. Combinations still numbers in hundreds per person. So basically this varies from hundreds to thousands in a person. The cataloged allele count keeps rising as more genomes are sequenced worldwide. So basically, if you consider the entire population of humans, you will see much variation there. Okay. Now, why is MHC very important? Because MHC is the reason why organ transplant is such a difficult procedure. Okay, we hear about organ transplantation and how difficult it is, and there is graft rejection that happens, and that is majorly because of your MHC. So compatibility testing or HLA typing. So compatibility testing is called HLA typing. Okay, before organ transplant checks exactly this diversity, and a poor match results in graft rejection as foreign, and what happens is the immune system gets turned up and it rejects the graft completely. Now, this is how the gene cluster looks like, okay? So the MHC region has three classes that we saw. First one is A, B and C, and it is called HLA, A, B and C. Then you have the second one. Second one is DP, DQ and DR. There are two segments of genes in each, alpha and beta in each, okay? Now we haven't talked about HLA3. That is because HLA3, class 3, this does not code for any MHC protein molecule. This codes for completely different protein molecules that are involved in the immunity, involved in immune responses, but they are not MHC molecules. Okay, this codes for three proteins, this C4, C2 and BF, which are involved in complement activation. Okay, they are immune response proteins, but they have nothing to do with MHC, these three. They also code for tumor necrosis factor alpha and beta proteins, and they also code for C proteins, which are again complement system proteins. Okay, there are other proteins that these code for, and they are necessary during the antigen presentation and processing, but they are not MHC. So we will read about that in the upcoming slides. Okay. Achha. So basically there are three genetic pillars behind MHC diversity. So how is MHC so diverse? We will look into that. The first one is polygenic. So you can see that there were multiple genes in the same locus, right? So multiple MHC loci exist per individual. That is HLA ABC for class 1, HLA DP, DQ, DR for class 3. So there are multiple MHC loci. And if there is a defect in one, that will compensate, the others will basically compensate it, right? And the second thing is polymorphism. Now we talked about alleles, right? So your MHC will be different from my MHC, and that is why graft rejection happens. So this means that the each locus has many allelic variants across the population. Okay, so no simple dominant recessive pattern is there, and every allele codes for a distinct protein. So what does this mean? This means that both the genes, so coming from the mother and coming from the father, right? So there is maternal and paternal allele, and both of these are expressing equally. There is not one dominant and one recessive in this pair. Both of them are co-dominant. Okay, both of them are expressing equally, and that is co-dominance. So that means both of them are important, equally important. Okay, so both maternal and paternal alleles are expressed equally on the cell surface, not recessive and dominant. So one infected macrophage can display all inherited variants at once. So all of these three properties, first is lot of number of genes are there, then there is polymorphism, and the second one is co-dominance. So all of this makes these MHC molecules very, very diverse. Okay, genetically diverse, and therefore in the protein sequence also there is a lot of diversity. Now, where the variation lies and where it comes from. So how is it that they are so diverse? Okay, how is it that so many different allelic forms are formed? Okay, so now see, this is your MHC molecule where the peptide binding cleft is there and the peptide is coming and fitting into it, right? This region is once again hypervariable. Remember hypervariable regions in antibodies? So this is also hypervariable region. That means that the residues cluster almost entirely in the peptide binding cleft. So this entire peptide binding cleft has huge variability between these MHC molecules also. Okay, so alpha 1 and alpha 2 of MHC1 and alpha 1 beta 1 of MHC2, they are very, very variable. Now, the regions that contact the CD8, that is alpha 3 and beta 2 microglobulin, or CD4, which are T cell co-receptors, they stay invariant, of course, because the CD4 and CD8 are same for all the T cells, right? And if this region has to contact this co-receptor, they will have to stay consistent. Okay. Now the result is that polymorphism widens the range of peptides that can be displayed without disturbing the T cell co-receptor binding. Okay. There you are getting the specificity, plus you are getting this antigen binding property also by this variations that is there in this peptide binding cleft. Now the origin. Now how is it that this is becoming so hyper variable, right? So that is because of something known as gene conversion and your normal, regular, conventional recombination. Okay. Now you know that during meiosis, what happens? This, let's say this is one chromosome and this is the other chromosome from mother and from father. These two come together and they form this chiasma, right? So a part of the chromosome, it basically forms this chiasma. You can see this reciprocal crossover. This figure is given here. And then what happens? Some genetic recombination happens. So in normal case of meiosis also genetic recombination is happening. And that is how the variation in the progeny is coming, right? We are not exactly 100% copy of our parents. We are separate individuals as of ourselves, right? And that is because of meiosis. So in normal meiosis, there is some variation that is getting added up. Besides that, there is something called gene conversion. So what is this gene conversion? So see, chromosomal misalignment happens during meiosis, and this swaps the short sequence blocks between similar genes and is the major driver of new alleles. So the figure is given here for you to understand what is gene conversion. So you can see that a small segment from this red uh, chromosome is now incorporated into this black one. And what is LOH? LOH is basically loss of heterozygosity. So now the two chromosomes that you can see, they are not distinct. There is a patch of one in another, and it is different from the ones that were there in the parents. And that is how with every cell division, some differences keep on adding up, and that is how we get a wide variety of genetic sequences and leading to different kinds of proteins. Okay, now we have this small MCQ. You can try answering this. Okay. Which of the following currently ranks the genetic contributors to overall MHC diversity in an individual? From the most fundamental to the most consequence amplifying. Okay, so try answering this. So we talked about three different things. Now, which of these will be most fundamental to the one having most implications? B. Okay, let's see which is the right answer. So basically, yes, very good. B is the right answer, but how is that happening? Let's see. Okay, so polygeny. First thing is polygeny, which provides multiple distinct loci. So there are multiple genes, first of all. Second thing is polymorphism. There are multiple alleles in different individuals of the species. And the third one is codominance, that ensures that in a heterozygous individual, both maternal and paternal alleles at every locus are expressed on the cell surface equally and simultaneously, rather than being masked or multiplying, rather than merely adding to total displayed diversity. Okay, and that is why from the most fundamental to the most consequence amplifying, these properties are. Now we come to MHC restriction. What is this? So basically, a T cell receptor must recognize the MHC molecule and the peptide. Now see, the T cell receptor, it is not that it is going to recognize only the MHC molecule and the peptide, it is going to recognize both. Okay, and together and not separately. Some peptides, so let's say that it is the same peptide but presented on wrong MHC, will lead to no recognition. So that is why MHC class 1 and class 2 are coming into picture, right? Because the co-receptor is there, then your TCR is there. All of these are recognizing exactly what type of MHC it is. Plus, it will also recognize the antigen presented on it. Okay. So, same peptide but presented on wrong MHC will lead to no recognition and no T cell activation. Right MHC but wrong peptide presented, once again no recognition and no activation. So this is why a transplanted organ or even a patch of skin is typically rejected within 7 to 10 days if the MHC is mismatched. Alright, so MHC sequence also has to be very, very consistent. Now there is something known as alloreactive T cell. So what is this alloreactive T cell? A small population, roughly, one second. Okay, yeah. So there is a small population of roughly 1 to 10% of the repertoire that does recognize non-self MHC directly. Okay, independent of the peptide presented, with very high affinity and serve as the basis of graft rejection during organ transplantation. Okay, so even if the peptide, the antigenic peptide presented is correct, if it recognizes the MHC, so there is a very small percentage of T cells which recognize the MHC molecule only, irrespective of whatever antigen is there, and these serve as rejection of graft, and they are called alloreactive because they are checking the allelic variants. Okay, now how is this happening? There is this very nice, I think this is a review paper in Nature, published in Nature. This is Nature and Cell. These are the biggest journals in biology. Okay, so there is something called allogenic cell, and this allogenic MHC is there, class 1 or class 2, and this is being recognized by this T cell, and this is shown in green. And the self MHC class 1, this is shown in pink. Okay, so initially there is this funnel. So initially there are multiple types of T cells. They can have affinity for this self antigen, they can have affinity for this allogenic cells and allogenic MHC. That means it is taken from external source, and then what happens, thymic selection happens, and you can see that most of them are removed from the body. If they are self interacting, they are removed from the body, but then in the repertoire there is some very tiny population remaining that are actually alloreactive T cells, and these are the ones that are preventing your grafts. Okay, Now we come to something called superantigen. Now what is a superantigen? Superantigens are antigenic peptides which can be from these different bacteria. An example is given, which is Staphylococcus aureus. Remember there was this, we had talked about this in our first and second class, that this is the bacteria which is responsible for all your pimples, okay, and all the skin conditions, boils and all. So there is this particular bacteria. There are also other bacteria, of course, and this particular bacteria releases some kind of enterotoxin which is called staphylococcal enterotoxin. There are other toxins, for example toxic shock syndrome toxins. These are some examples, and these toxins, the property of these toxins is that they bind directly to the MHC alpha 1 domain and the TCR VB domain outside the normal peptide groove. Okay, so no antigen processing is needed, and they force the MHC and TCR together even without a matching peptide. So that is the property of these superantigens. And the result is that broad non-specific activation of an entire T cell population happens, and not just antigen specific clones. Okay. Now what happens? So several viral proteins can also act in this way. Okay. Now this has been used in therapy very intelligently by us humans. So what happens is, in the primary vaccine dose, this antigen, this super antigen is clubbed with our desired antigen, and it is introduced into the patients. Now what happens? Initially, a larger batch of immune cells are getting activated. Normally what happens? T cell response, it takes a lot of time, right, to activate the specific T cells and B cells. Here what happens, since the T cells, even without the peptide, they are getting activated. So what happens? These super antigens are mixed into a primary vaccine dose to broadly prime the immune system. But then what happens in the secondary, in the booster doses, then what happens? Specific T cells have already formed, and we don't want to introduce this super antigen anymore, because they are very, very specific T cell receptors and B cell receptors. These have been developed, and we want the memory T cells and B cells to be there. And that is why only your specific antigen is introduced in the booster doses of the vaccine. Okay, so that is how this works. Now we have a small MCQ. Try to answer this. Which of the following best distinguishes super antigen mediated T cell activation from conventional antigen specific activation? Okay, super antigens require processing by proteasome before MHC loading. B. Okay, super antigens bind outside the peptide binding groove, cross-linking MHC2 alpha 1 and TCR-VB regardless of peptide specificity, activating a much larger fraction of T cell pool. Yes, correct. So that is the right answer. Good. Okay, now we will move on. Now we will come back to class 1, 2 and 3 MHC. Now remember, I said that this class 3 is a very strange class of MHC molecules, because they do not code for any MHC proteins. And that is why class 3 MHC is called class 3 MHC, only because this is there in between class 1 and class 2 MHC. So it is there in the same genetic cascade. That is why it is called an MHC class. Otherwise it wouldn't have been called. Okay. So human chromosome 6 arrangement. Class 3 region lies in between class 2 and class 1 region. Class 2 is not an antigen presenting molecule at all. It is a catch-all region of immune support genes. Now, this region includes complement genes. I told you, complement system proteins are formed because of this, and cytokine genes, for example, tumor necrosis factor alpha and beta. Some proteins involved with antigen presenting processes, for example, TAP1 and TAP2. Now we will talk about TAP1 and TAP2 in the upcoming slides. Okay, these are actually very important. We will see their role in the upcoming slides. So this TAP1 and TAP2 are basically transporter proteins. They are associated with antigen processing at different stages. Okay, and then there is something called LMP, which is low molecular weight protein. These are also important units inside the proteasome, and these are the genes that are there in class 3. So this is because of its position that it is called class 3 MHC, not because of its function. Okay. Acha. Now we come to something very important, which is antigen presentation. The two pathways of antigen presentation. One is called endogenous pathway, which is for the MHC class 1, and there is MHC class 2, and it is exogenous pathway. Okay. Now the source of the antigens is, for the endogenous pathway, it is viruses, viral proteins, tumor antigens, defective ribosomal products. So inside the cell, what happens is, see, inside the cells, constantly proteins are getting produced. Now, if you know, there can be a lot of stress condition inside the cell. For example, let's say you have been scrolling on your phone till 2 o'clock, 3 o'clock at night. Then what happens? There is a lot of stress that is going on in your body. Or let's say there is exam pressure and you are dehydrated and you're having this headache. That means there is low oxygen condition in your brain, right? So all of these things result in misfunctioning inside your cells. Now the cells are producing proteins not in a very like optimal way. So what happens? A lot of misfolded protein products are produced, a lot of truncated protein products are produced. Then what happens? In the normal functioning of the cell also, proteins are produced, but then proteins get degraded. Okay, so it is not like some protein will be there forever. There are housekeeping genes also which make proteins, but they also get degraded and they also get formed again. Okay, so there is a shelf life for every protein. Now these worn out proteins, for example, they have worked and they are supposed to die. So these proteins, they also undergo lysosomal degradation, right? So what happens is this now we come to peptide length. So these are also like supposed to undergo this lysosomal degradation. Okay, so we come to peptide length. It is 8 to 10 amino acid. It is relatively shorter, and they are processed by this ubiquitin-proteasome system in the cytosol. So what is ubiquitin? Ubiquitin is a very small protein, very, very stable protein. This protein is attached onto a protein that will undergo degradation. So this is like a marker for a protein that has to undergo degradation. And multiple ubiquitin molecules are attached onto the same protein, okay, tagging it for degradation. Now, these peptides are basically presented to CD8 plus cytotoxic T cells. Okay. Then we come to the second one, which is exogenous pathway, and the source is extracellular pathogen. You have eaten something, you have eaten, let's say, some very cheap uh, food stall food, okay, and exogenous pathogens have entered into your body. And then what happens? There can be pathogens that have entered, like for example, mycobacterium. If this has entered, you're dead, okay. So there can be pathogens basically entering into our body from the food that we are eating, from the water that we are taking in, from the air that we are breathing, from everywhere. And the peptide length is around 13 to 20 amino acids processed in acidified endosomal or lysosomal vesicles. And then what happens? It is presented to CD4 helper T cells. Okay. Now we will look at this in a lot of details now. Acha. So this entire thing is once again shown here for type 1 antigen presenting pathway. You see there is this endosome. Endosome merges with the lysosome to give your phagolysosome. Okay, or you can also call it phagosome and then lysosome, and so phagolysosome. And then what happens? They are degraded, peptides are degraded, they are presented on the surface. The other one is there are these, one second. Yeah, so there are these vesicles inside which the processing happens. Okay, in the second case. Now, this is the entire picture. Okay, this is the entire story. So let us go through this once, because if we understand this, then the rest of the slides is actually going into even more depth. So then you will be able to catch up properly. All right. So we'll try to understand this first. Let's see what is there. First look at this endogenous pathway MHC class 1. So there is this endogenous antigen that has been produced inside our body. Endogenous antigen is degraded by the proteasome, remember. Okay. And what happens? Peptide is transported to the RER, that is rough endoplasmic reticulum, via TAP. Remember we talked about TAP. I said TAP proteins will come up in the upcoming slide. So this is your TAP. Okay. So this TAP protein is taking it to the rough endoplasmic reticulum. Then what happens? Class 1 MHC alpha chain binds to calnexin. Now this calnexin is it is a protein which is helping to, it's a chaperone. Chaperone is also a protein, okay. So this is a chaperone protein which is helping to stabilize this MHC. Now if you remember, MHC molecules, they need to be bound to this peptide to remain stable. Otherwise what they will do is they will either bind to any random peptides or they will undergo degradation, because these proteins are not very stable, okay. So what happens, there is this chaperone called calnexin. This binds to this MHC class 1 alpha chain. Then what happens? Beta 2 microglobulin comes and then replaces this calnexin. Then another chaperone comes. It is called calreticulin. There is another protein which is an adapter protein. This comes and its name is tapasin. These come and bind. All of them form a complex. Okay. Then what happens? MHC captures the peptides and then the chaperones dissociate. And MHC is now relatively stable because it is bound to the peptide. Now class 1 MHC peptide is transported from rough endoplasmic reticulum to the Golgi complex and to the plasma membrane. And there it is displayed on the surface of the plasma membrane. Okay, we will look into all of these steps, okay, in a lot of details. Now we come to the second one. Acha, remember there is a crosstalk between the two. Okay, we will talk about that later on. Okay, so class 2 MHC alpha and beta bind invariant chains. Now here there is a different story happening. There is another chaperone called invariant chain. Okay, this is also a protein chaperone. Now this one MHC class 2 is also not very stable. Okay, and this MHC class 2, this one actually binds to this invariant chain chaperone. This invariant chain has a very small region. We will call it clip. Okay, let's say this is the MHC class 2 protein and it comes, and this clip region comes and binds to this peptide binding cleft. The rest of my hand is invariant chain. Okay, this clip region comes and fits here. Why? So that this peptide binding cleft does not bind to any random peptides inside the cell. Let's say any self peptides inside the cell. Okay. Now MHC complex is routed through the Golgi to endocytotic pathway and compartments. Invariant chain is degraded. Now in the endosome, you know that there is very high pH and there is degradation of these peptides that are happening. Okay. So degradation of this invariant chain also happens. Clip region remains bound so that no peptides, random peptides come and bind. Okay. Clip region now has to be removed, and that is done by a lot of proteins. Okay. Now exogenous antigen is taken up. Okay. So invariant chain is okay. Now see, exogenous antigen is taken up, degraded and routed to endocytotic pathway compartments. Then something known as HLA-DM. This one comes and mediates exchange of clip for antigenic peptide. Now when this particular protein comes, clip is removed and the antigenic peptide is able to bind. Then something called, so there are many, okay. There is something called HLM-DO. There is something called HLA-DM. We will talk about that in details. Now see, after that, the peptide with very high specificity and affinity comes and binds, and then it is taken and it is presented on the surface of the cell. Okay, so this is the entire story of MHC class 1 and class 2. Now we will go into the details of this. Achha. So MHC class 1 we will look at first, and there are multiple steps. Okay, so first thing is tagging. Tagging, if you remember, I said that ubiquitin will be added in the form of tags to a protein that needs to undergo degradation in the proteasome. Okay. So, defective or unneeded cytosolic proteins, 30% of newly synthesized proteins which are misfolded, truncated, non-functional, these proteins are called DRIBs. So DRIB is a defective ribosomal product, and the remaining worn out proteins. Worn out proteins are the proteins whose shelf life is over. They have done their work. They now need to get removed. Okay. So they are marked with polyubiquitin chains. Polyubiquitin means multiple ubiquitins are sitting on top of one another. Now there is something very important which you must remember, that in a lot of signaling, cell signaling, when you read about it, you will see that ubiquitin is coming and binding to a protein. Okay, but not necessarily every time ubiquitin binds, a protein is marked for degradation. That is not the case, because ubiquitinylation is a post-translational modification. It happens in proteins, even normally during their normal function. Okay, so polyubiquitinylation is a specific marker. So ubiquitin upon ubiquitin upon ubiquitin, that is a marker, but then ubiquitin sitting on the protein, that is not a marker for degradation. So remember that. Achha. Now proteins are covalently tagged with ubiquitin in the process of polyubiquitination, and they are delivered to the proteasome. Now what is this proteasome? This is a 28 subunit long barrel. So inside that barrel, the protein is unfolded and it is cleaved into short peptides. Now in the proteasome, something happens when there is immune activation going on. When there is interferon gamma signaling, what happens? These three proteins, LMP2, LMP is low molecular weight protein. Remember, so LMP2, 7, and there is something called MECL1. Please note down the full forms. Okay, I have not mentioned them in this slide, but please remember the full forms. Okay, now these subunits replace the standard subunits in the proteasome to generate MHC-1 compatible cuts in the peptides with C-terminal hydrophobic residue for MHC-1 binding. So, what happens is, let's say this is my peptide. Now, if you remember, MHC-1, in the peptide binding cleft, it is relatively narrow, so shorter peptides can get in. And also the way these peptides bind, that is also very, very sequence specific. Not completely sequence specific, but it is in such a way that the last residue, the C terminal, should have hydrophobic amino acid, and at the center also there is some kind of hydrophobic interaction between the peptide binding cleft and this MHC class I. So it is relatively more specific towards the sequence of this peptide. Now these peptides that we have, they need to be cut in such a way that they will be able to go and bind to this peptide binding cleft, and that is done by this particular complex. Okay, now this particular complex ensures that the peptides are cut in such a way that their C terminal will have hydrophobic residues needed for MHC-1 binding. Achha. Now we come to TAP1 and TAP2. So TAP is transporter associated with antigen processing. This is its name. So TAP1 and TAP2 form a heterozygous complex, and then what happens is an ATP binding cassette or ABC transporter. So ABC transporter is kind of a channel on the membrane, and this allows the peptide to go from the cytosol into the ER by active transport. So what is active transport? Active transport means that ATP is needed for this, and it is going from lower concentration to higher concentration, right? That is why ATP is needed. It is a active transport basically. Okay, and that is done by this ABC transporter known as TAP1, TAP2. Then within ER, Achha. So this transport is mediated by these proteins TAP1, TAP2, and then through this ABC transporter, it is basically going. Okay. Now there is something called trimming. What is trimming? Now inside the ER or endoplasmic reticulum, there is something called ER aminopeptidase, which is an enzyme which trims the oversized peptides into final 8 to 10 amino acid length. ER chaperones, calnexin. Calnexin is the chaperone which binds to this MHC class I molecule, right? So to ensure that it is stable. And then calreticulin and ERP57 plus tapasin. Tapasin is a adapter protein. All of these come together to form this peptide loading complex or PLC under high affinity until a high affinity peptide comes and locks in. So here is also there is a selection process going on. So multiple peptides can come, but if they are low affinity peptides, that means they are not having very high specificity and affinity for this particular MHC class I. They are not bound to this and they are removed. And only when the high affinity peptide comes and sits on it, then it is locked, and it all these other proteins, calnexin, calreticulin, etc., these are all removed, and then this complex is exported via Golgi through your transport vesicles to the surface, and for 2 to 4 days it is sitting there and waiting for cytotoxic T lymphocyte to come and recognize it. Okay, that is the entire process of this MHC class I. Now, to understand this visually, you see these endogenous antigens are there. Ubiquitin comes and sits on them, and basically what happens is it is taken for degradation inside the proteasome, and there it is degraded into peptides. Now peptides can either be carried by tap into endoplasmic reticulum, and inside they will meet MHC class I, which was bound to the chaperones, and now it is binding to this MHC class I and it is getting transported to the plasma membrane for presentation to the T cells, cytotoxic T lymphocytes. Coming to the endocytotic pathway, here what happens, exogenous antigens are coming in, they are taken in by endocytosis or phagocytosis, and then it is going into the endocytotic compartments, and then the peptides are getting formed through acidic environment and cleavage, and then this is going to the peptide class II MHC complex. Now if you look at the figure on the right, you will see that this ubiquitinating enzyme is there, and there are 3 enzymes which are involved in this ubiquitination, and then what happens, ubiquitin group gets added to our peptide through covalent bond, and then this peptide is carried into the proteasome. Proteasome looks like that, a long tunnel barrel kind of thing. There it is cleaved into peptides. Okay, now we will look at the next slide. So this entire thing that we talked about, it is explained here once again. So see, surveillance of intracellular threats, that is viral infection, tumor, neoantigens, and harmlessly self proteins. All of these undergo this entire procedure, and let's see what this is. So the protein pool is there. So the worn out proteins up to 30% of defective ribosomal products or DRIPs are there. They are ubiquitinated. So polyubiquitination tag is added. It is called the kill signal for degradation, and then it goes into the proteasome, and the proteasome is having these subunits which are heptameric. So there are 7 alpha, then 7 beta, again 7 beta and 7 alpha. So basically 28 subunits are there, and this is also having this catalytic code and all that. And then what happens, these unfold and cleave the proteins into the shorter peptides, and then TAP is helping it. So TAP1, TAP2 pumps peptides across the ER membrane, and it enters into the ER lumen, and there ERAP comes and this trims. So ER aminopeptidase. So this trims into the ideal length for MHC class I. Then what happens? Then, okay, so proteasome architecture is once again explained here, okay, for your understanding. And immune protease swap is very important. Here what happens, whatever standard proteasome subunits were there, they are replaced by this LMP2 and these other proteins. And then what happens is, in presence of interferon gamma, 3 standard catalytic beta subunits are swapped for LMP2, LMP7, MECL1, shifting the cleavage to favor C-terminal hydrophobic or basic residues. And this is optimized anchor residue ready for MHC class I. Okay, so anchor residue means the residue that is going to come and bind to this peptide cleft. Okay. Now, we go to this chaperone assembly. So remember what was happening? MHC class I was there. MHC class I is not very stable by itself. It is binding to alpha, your calnexin, which is a chaperone. So MHC class I alpha chain is binding to calnexin. Then what happens? Beta 2 microglobulin comes and calnexin is removed, but then tapasin, which is an adaptive protein, calreticulin, which is another chaperone, these two come and bind, and this entire complex of calreticulin, tapasin associated with MHC class I, these together plus the beta 2 microglobulin, these together make the this complex, right? And then what happens? Peptide molecule comes and this peptide loading complex is dissociated, and the specific and high affinity binding peptide comes and enters into the um, peptide binding cleft, and it is selected and it remains bound. Rest of it dissociate and it exits the rough endoplasmic reticulum and goes to the cell surface for display. Acha. The same thing is explained once again here. So alpha chain plus calnexin. So alpha chain is synthesized, calnexin stabilize it while it awaits the beta 2 microglobulin. Calnexin is released and still the peptide free dimer is okay. So MHC1 is unstable until a peptide occupies its groove. Okay. Folding is chaperone dependent at every step. Now see what happens. This is passed to, so this peptide free dimer is passed to calreticulin plus ERP57. This is another protein. Then what happens? Tapasin bridges TAP. So tapasin forms the peptide loading complex positioned at the TAP peptide entry site, and the peptide is locked in. So high affinity peptide binds and the complex is stabilized and dissociates from the chaperone. Then what happens? Surface display. So it goes from the Golgi through the trans-Golgi network into the vesicle, which is carried to the plasma membrane, and then it is recognized by the CD8 plus T cells. And the peptide loading complex is made up of calreticulin. Remember these details, okay? These are important. Calreticulin, ERP57, tapasin, and TAP holds the MHC1 at the peptide entry site until the high affinity peptide locks in. Now for 2 to 4 days it is sitting on the cell surface until your CTL is coming and recognizing it and binding to it. Acha. Now this entire thing is once again shown here. So this is how TAP looks like. TAP is this ABC transporter, okay, which is sitting on the rough endoplasmic reticular membrane, and it uses ATP for this entire process. Now once again the entire process is shown in the form of a diagram in your right hand side figure. So this is your proteasome. It is coming in and it is going inside into the ER, and then all of this assembly is happening on the membrane, and then it is getting transported by a vesicle. Okay. Now we have cross presentation for covering the gaps. Next slide, we have MCQs. Okay. Now see, the problem is that only 3 cell types can activate toxic cytotoxic T cells. They are dendritic cells, macrophages, and B cells. Some viruses never infect these antigen presenting cells directly, so the endogenous route is unavailable. The solution is that dendritic cells and macrophages, they phagocytosize virus infected dead cells, and the viral antigen escapes the phagolysosome into the cytosol and is treated exactly like any other cytosolic protein. So it re-enters the standard proteasome TAP MHC1 route called cross presentation, and this guarantees that a cytotoxic T lymphocyte is always going to recognize it, even the pathogens that cannot infect presenting cells directly. Okay, right. So this is once again given here for your understanding, and we will come to the questions. So, which combination correctly matches the MHC class I peptide loading complex components with their primary function? Try to solve this. Calnexin trims the peptide length, tapasin degrades. Acha, B. Let's see which is the right answer. Yeah, so B is the right answer. Good. So basically, calnexin holds the chain before beta 2 microglobulin binds. Remember the names. Right now, you guys are answering very fast. That is very good. But then, please don't get confused later on. Don't forget later on. Okay, revise again and again. Okay. So yeah, so that is the whole picture, and that is what you need to remember. Okay, good. Now we have the next question. The immunoproteasome differs from constitutive proteasome by replacement of which of the subunits in the what is the functional consequence? Right. Why are all the answers B? You guys are okay. So interferon gamma induced swapping is actually happening, and that is how you're getting this specificity where it is going to cleave at the hydrophobic or basic C termini. And that way the peptides will be able to bind to the cleft. Now, the genes encoding TAP1 and TAP2 and LMP proteasome subunits are located in. Exactly. Yes. So they are located in the class 3 segment of the MHC. Okay. Now we come to MHC class 2 antigen processing. Okay. So in MHC class 2, what happens? There are two different streams. Once again, one is the antigen and the other one, of course, your MHC molecule. So two come and converge at a point. Okay. So what is happening? Let's see. The peptides come from outside the cell through phagocytosis, endocytosis, and macropinocytosis, or under stress, self-material via autophagy. Okay. Now, engulfment, phagosome or endosome mediated engulfment. Now, see, phagocyte mediated engulfment. So, extracellular material taken up by phagocytosis, receptor mediated endocytosis, or macropinocytosis. Phagolysosome gets formed. So, phagosome or endosome fuses with the lysosome. Inside the lysosome, it is very, very acidic. The protease-rich environment progressively degrades the protein, and these small peptides are formed. Now, in the second stream, what is happening? Your MHC2 alpha beta heterodimer is synthesized. Once again, it needs to bind to this peptide to remain stable, otherwise it is unstable. So what happens? Unlike MHC1, this remains unstable alone. Okay. Now, something called invariant chain. Invariant chain is written as II. Okay, here. Now, invariant chain is once again a chaperone which comes and binds to this MHC class 2 molecule. There is a very tiny segment of this invariant chain which is called clip. Clip comes and enters into the peptide binding groove and binds to it. So clip occupies the groove and the three pairs form a nonamer. Nonamer means 9. Okay. So invariant chain, clip and this MHC2. 3 together form a nonamer. Now see, this is diverted from secretion. So that means its trafficking signal routes the complex to the endocytotic or lysosomal system. And that is where it comes in contact with the antigen. Okay, what happens when it comes in contact with the antigen? We will see that. Right now, let's look at this in a little more details. So this MHC class 2 has this invariant chain and clip. Okay. Newly synthesized MHC2 alpha beta is unstable on its own. It is stabilized in the ER by a chaperone protein called invariant chain. The segment of the invariant chain called clip. Clip is class II associated invariant chain peptide. Physically occupies the peptide binding groove, keeping it stably blocked and folded correctly until the complex reaches the endocytotic pathway. 3 MHC, 2 alpha beta heterodimers, each paired with invariant chain, further assembles into a complex monomeric complex for trafficking. Okay, so 3 MHC2 and invariant chain complexes come together to form this monomeric complex, and this invariant chain actually carries the signal peptide to the endosome. What is signal peptide? If you remember, we were talking about this in the previous class. So this entire path of a particular protein, this is determined by a sequence of amino acids which is extremely specific, whether a protein is supposed to remain in the cytosol, whether it is supposed to go to the mitochondria, whether it is supposed to go to the cell surface or Golgi or nucleus. That is determined by a tiny stretch of amino acids, either in its N-terminal or in its C-terminal, mostly N-terminal. Okay, that is called your signal peptide or a localization signal. Okay, for example, there is this nuclear localization sequence. Then you have, for a protein to come into the ER, it will have this signal peptide. Okay, for it to go into the membrane, cell membrane, it will have a signal peptide. That is what the other proteins recognize and take this particular protein to wherever it wants to go. Okay, now you have this 3 MHC2 II pairs trimerize, keeping the complex intact while it traffics to the endosome. As the vesicle acidifies, proteolytic enzymes activate and they cleave off the II. Okay, only the clip is left. Then what happens? Only the small clip fragment remains lodged in the peptide binding groove afterward. The cliff loaded MHC2 vesicle then fuses with the vesicles carrying processed exogenous peptide fragments. Okay, and they are now going to come into contact. So see what happens. They are basically merging. The two vesicle streams converge, internalized antigen meet the MHC2, and then the clip is exchanged for the best fit, peptide. Now, this is called MHC Class II compartment or MIIC. That is where this merging happens. Now, how this merging happens? There are some proteins involved in this merging process also. For example, your clip has to be removed by a protein called HLA-DM. What is this? Catalytically destabilizes. So this HLA-DM, this protein comes and it catalytically destabilizes clip and it opens the groove for this pool of antigenic peptides to come and bind. Now the low affinity antigenic peptides, they are not allowed to bind. Peptide editing, okay, HLA-DM, this releases the low affinity peptides and high affinity ones lock in and resist exchange. HLA-DO halts it. So this is a negative regulator of HLA-DM. So HLA-DM will make the peptides move out, but HLA-DO will stop the right peptide from moving out. Okay. Now negative regulator of HLA-DM ends further exchange once the loading is optimal. Then what happens? This complex is sent off to the plasma membrane and it is once again sitting there for 2 to 4 days for recognition by helper T cells. So the clinical correlation is that HLA-DM mutations impair antigen processing, revealing its essential catalytic role. But HLA-DO deficiency gives a much milder phenotype consistent with fine-tuning role because it has this fine-tuning, right? Okay. Now, whatever we have talked about, that is given in the form of a figure. So there is this MHC class II molecule with alpha and beta chain. Invariant chain is coming. Invariant chain is having this tiny clip molecule. Clip molecule is entering here, sitting. Invariant chain is also sitting here. Then what happens? the invariant chain gets degraded in the proteasome and the clip molecule is released by HLA-DM. HLA-DM is also like an MHC molecule. It is looks very similar to that. Then your HLA-DO comes. Please see the full forms. Okay, please see the full forms. So HLA-DO comes and HLA-DO ensures that the HLA-DM activity is stopped and the right peptide, once the right peptide comes and sits, the HLA-DO comes into play and it ensures that HLA-DM is released from the scene and this entire complex is now shipped outside the cell membrane. So peptide editing, HLA-DM and HLA-DO. HLA-DM is an MHC-like molecule with no peptide groove of its own. It destabilizes the MHC2 clip interaction. Clip falls away, leaving the groove open and briefly vacant. Candidate peptides from the fused vesicle compete for the open groove and poor fit peptides are exchanged out while the DM remains bound. HLA-DM remains bound. Okay. Now the best fit high affinity peptide locks in and it is stable enough to survive for 2 to 4 days en route to and within the lymph node. So HLA-DO is a negative regulator and then displaces the DM once the loading is optimal and stopping further exchange. Okay, that is all about this peptide MHC class II antigen processing and presentation. Okay. Now, as I told you that every protein in our body, none of the proteins are there forever. The proteins are expressed when they are needed and they are degraded when they are not needed. Okay. Same with MHC molecules. Without infection, the E3 ligase March1 continuously ubiquitinates MHC2, sending most of it for degradation. Only a small baseline amount of MHC2 reaches to the cell surface at rest. Infection triggers TLR signaling. TLR is toll-like receptor, which recognizes your PAMPs, right? TLR signaling, that is bacterial LPS engaging TLR4 to be even more specific, which halts new March1 transcription. So there is a regulation of both March1 and regulation of MHC2 here that we are seeing. Okay. So March1 protein itself has a short half-life of 30 minutes. So existing March1 is rapidly cleared. With degradation switched off, surface MHC2 rises sharply, boosting antigen presentation exactly when it is needed. Achha. Okay, so before we go to this MCQ molecule, I'll tell you something very interesting. Okay, so you can see that proteins inside our cells, they get degraded within a few minutes, right? Or maybe a few hours at most. So the thing is that when these proteins, you know that industrially important proteins, they are often lyophilized. So lyophilized means that whatever water was there in this solution, it is removed and the protein is frozen. Okay, and it is preserved for in the form of a powder, and a powder can stay intact for years, right? And even if the protein is not lyophilized, even if the protein is kept in solution form, as industrially important proteins, they need to be very, very stable, right? And the protein here, see what is happening, all of these proteases are there which are coming and cleaving the protein. But when you are working with this protein in the lab, proteases are not there because you are adding protease inhibitors. These are again separate chemicals. Okay, they are added so that the proteases cannot act. Okay, so there is a full branch of science called protein engineering, which changes the sequence of naturally found proteins which are of industrial importance to make them more resistant to this proteolytic enzymes, more resistant to temperature, and have longer shelf life. For example, I can give you an example here that if you know about this PCR, polymerase chain reaction, there at 95 degrees centigrade, your thermally very stable, thermally stable DNA polymerases are used. For example, PFU polymerase. Okay, so these are used, they are taken from these thermostable bacteria, archibacteria, which are found in hot springs, and like that is also naturally found, and then more mutations have been done on them to make them highly fidel, to increase their fidelity so that they don't make mistakes while making these new DNA molecules. And so basically that is one way of seeing it, but there are multiple ways. Now there are computational tools, now there are experimental tools in which you can basically engineer proteins. So yeah, that is it. Our lab works with this, and I just told you this as a story. Now try to answer this question. In the MHC class II pathway, HLA-DO functions as a chaperone that stabilizes the empty MHC2 groove before the clip binds, catalytic peptidase that cleaves the invariant chain, a negative regulator of HLA-DM, restricting the further peptide exchange once optimal landing occurs, and an ATP-driven peptide transporter analogous to TAP. Yeah, so not all answers are B. Come on. You'll have to read the question at least. So it is C. C is the right answer here. Okay, HLA-DO is a negative acting negatively to HLA-DM, and it is restricting further peptide exchange once the right peptide comes and binds. Okay. Now, which statement about the invariant chain and clip are incorrect? So, II occupies the MHC peptide binding groove and clip segments via its clip segments during the ER transit. II provides trafficking signal that routes MHC2 to the endocytotic pathway. Clip is removed by HLA-DM independent spontaneous dissociation at neutral pH. Proteolytic cleavage in the acidified endosome degrades most of II, leaving only clip attached. D. Yeah. Okay. Let's see which is the right answer. Oh, I am sorry. This wouldn't be the right answer. So it is not neutral pH. It is definitely acidic pH. Okay. So sorry, one second. Yeah. So your right answer would actually be D here. Okay. So D would be the right answer. Now, clip removal is not spontaneous, and it specifically requires the catalytic action of HLA-DM to destabilize the clip MHC2 interaction. Without HLA-DM, the clip remains bound, and MHC2 cannot efficiently be loaded with the antigenic peptide as demonstrated in HLA-DM deficient states. Okay, and this wouldn't be neutral pH, it would be your oh, sorry. Which is incorrect. Oh, acha. So this is of course correct. Okay, that is why I was thinking that B is also correct. Okay, so which is incorrect? Incorrect is C. Incorrect is C. Okay. Acha. Right. Now, from infection to adaptive response. So, what happens is now we will look at the bigger picture. Okay, since day one we have been talking about the barriers. Skin, mucus, they form the first line of defense against invading pathogen. Still the pathogen enters, then what happens? The innate response kicks in. Neutrophils, monocytes and mast cells, they trigger inflammation, and you have this heat, pain, redness and swelling, which are the four hallmarks of inflammation. Then what happens? All these antigen presenting cells, they engulf this particular pathogen, and they bring this to the lymph nodes, and they present the pathogen for B and T cell activation. So antigen presenting cell carry antigen from the infection site to the nearest lymph node. Then what happens? Cytosolic antigen, that is MHC1, mainly dendritic cell. Exogenous or vesicular antigens, MHC2, presented by macrophages and dendritic cells. And then cytokine signaling happens. So cytokines and chemokines recruit and activate nearby T cells and B cells, and that is how they get activated. Right. Now the two signal rule for lymphocyte activation. This we have talked about in our previous classes. If you remember, there was a signal 1 which is binding of the TCR with the MHC class 1 or class 2, and you have this co-receptor binding. Those are two signals plus cytokine activation. This also happens. So there are two signals. Okay. So signal 1, TCR engages the peptide MHC on the antigen presenting cell. Signal 2 is cytokines released by the same activated APC antigen presenting cell plus the co-stimulatory receptor ligand pairs such as CD28 and B7. Both signals must arrive together. Yeah, sure. If you have a question, you can ask me.
So, ma'am, what is the meaning of the signal rule?
What is the meaning of two signal rule? Oh, there
Yes,
are
ma'am.
two signals. So basically this is a rule where two signals have to be on. So basically the TCR has to recognize this peptide and MHC complex plus co-stimulatory T cell receptor has to recognize this MHC molecule plus all this cytokine has to be released from the T cell. If there is only one signal, then what happens? The cell is going into energy. Okay, that we will talk about this in the upcoming. So this is the slide. Okay, so we are going to talk more about this. Don't worry. Okay, let's see what happens when there is only one signal. So the two signal rule means that two of the signals have to be coming for the like the rest of the process to go on. For the activation of the T cells, two different interactions have to happen. That is your two signal rule. Okay, so now both signals must arrive together from the same antigen presenting cell. If only signal 1 comes, then it leads to energy. Okay, now the result is if both the signals come, then what happens? Then what happens is proliferation into cytotoxic, that is from dendritic cell MHC1 presentation or helper effective T cell from MHC2 presentation forming Th1, which is T helper cell type 1 macrophage activating, and T helper cell type 2, which is B cell activating. T lymphocytes are differentiated. Okay, so basically a naive T lymphocyte gets these two signals and then they recognize this peptide antigen bound complex and they will recognize this co-receptor and cytokine. Under the activity of this cytokine, it is going to differentiate into Th1 and Th2 cells. Okay, for B cell activation, you know that B cell can directly bind to the antigen or it can also be stimulated by helper T cell. So B cell receptor, which is membrane antibody, it binds to the antigen directly, and the second signal is cytokine help from an activated follicular helper T cell, TFH, right? Both of these have to happen, plus of course your co-receptors will be involved. Co-receptors will also play a role there. And then what happens? Signal 1 alone leads to energy, and both the signals when they are coming, then differentiation into antibody secreting plasma cells and memory B cells happen. Initially all the B cells are IgM, later they are class switched into IgA, IgE and IgG. Okay, under continued T cell help. This you know, this we have talked about in a previous class. Okay. So epitopes and polyclonal response. Now coming to B cell immunity, immunity. You remember what is an epitope, right? So there is this antigen, this specific part of the antigen which interacts with the paratope of the antibody. This tiny part is called epitope, right? So epitope is a specific part of the antigen actually recognized by an antibody or B cell receptor. Now this epitope, this can be, now this is very interesting, this is very different. Now so B cells, they can recognize peptides in a very different way from T cell receptors. Okay, let's say that so let's say that this is the epitope and this is coming out this way. It can be present in the form of a linear peptide and the BCR will recognize it. It can be present in the form of this circular peptide, let's say, And um, let's say that the BCR, and in this circular peptide, you know that the proteins, they are always 3D, they are in 3D folded form, right? So in that 3D folded form, let's say there are two loops like this in this, let's say, heart shaped configuration. There are two loops, and both of these are recognized by this antigen, and this region, they are structurally very close by but sequentially very far away. Okay, but their 3D configuration is such that both of these are recognized by the same FAB region of an antibody. Then it is called conformational epitope. Okay, so we will look at this in details now. But when you come to the TCR, it is a cleaved peptide, small peptide. So they are always sequentially very, very close. Okay, TCR only recognizes the antigens that are the residues that are sequentially very close because they are one small cleaved peptide. But BCR can recognize conformational epitopes of antigens as well. Got it? Okay. Now, a continuous stretch of the primary amino acid sequence is a linear sequential epitope. Conformational epitope is discontinuous residues that are brought together only by the protein's 3D fold, and it is lost on denaturation. So when the protein is denatured, that means initially the protein was having this structure. When I am denaturing it, it is becoming long linear form. And then what happens? Its epitope is no more active because the conformational rearrangement has not happened there. So the antibody, the BCR, will not recognize this anymore. Okay. Unlike T cell, and remember that denatured proteins, they can be renatured again when it is temporary denaturation. Okay. Now, unlike T cells, which always see chopped linear peptides, B cells can reorganize the whole folded. They can recognize the whole folded antigen directly. Okay, one antigen with multiple epitopes activates distinct B cell clones, and this is called a polyclonal antibody response against the same target. Now we will talk about monoclonal and polyclonal antibody made in mouse and rabbit in one of our upcoming classes. Okay, so that is it. And the key takeaways for today's class is MHC diversity rests on three pillars. One is your polygeny, polymorphism, and dominant expression with gene conversion. T cell obey MHC restriction, okay, and MHC class 1 and class 2 run on two separately large non-crossing processing pathways, feeding CD8 plus and CD4 plus T cells respectively. Then you have your cross presentation. Let's dendritic cell activate CD8 plus cells even against pathogens that were never directly infected by, because they have obviously engulfed it. And then the peptide loading is tightly chaperoned and edited. Okay, leading to guarantee a stable and high affinity complex, and both B and T cell activation requires two independent signals, a built-in safeguard against autoimmunity. Now we have a lot of MCQs that you have to answer. Okay, so cytotoxic T lymphocyte fails to recognize a virus infected fibroblast that is clearly displaying the correct viral peptide on MHC class I. What is the most likely explanation assuming the peptide sequence is correct? B. TCR is restricted to a different MHC allele than the one expressed on the fibroblast. Let's see which is the right answer. Yeah, so right answer is that. Okay, this describes the MHC restriction. So TCR must simultaneously recognize both the correct peptide and the correct MHC molecule. Okay, and in this case, this is an allelic mismatch that is happening. Okay, a mismatch MHC allele, even displaying the exact correct peptide, will not be recognized. And okay, the other options are of course wrong. You can go through them. Now, a researcher observes that alloreactive T cells can recognize non-self MHC molecules even in complete absence of the peptide normally required for TCR engagement of self MHC. This phenomena is best explained by. Okay, let's see which is the right answer. So the right answer is A. This is cross reactivity of the TCR with an unusually large number of self-peptide or self-MHC combinations, one of which structurally mimics the non-self MHC and any bound peptide. Okay, that is why the alloreactive T cells can recognize non-self MHC as well, even in complete absence of peptide, normally required for TCR engagement. And remember, this is okay, this is only like less than 10% of your total T cell population. Okay, now during B cell activation, linked recognition refers to the requirement that B cell receptor and TCR must recognize physically linked covalently joined epitopes on the same peptide. Helper T cell provides signal to must be specific for a peptide derived from the same antigen that the B cell internalized via its BCR. Yes. So B is the right answer. Linked recognition means that two different signals are coming at the same time and both are because of the same antigen. Okay, one is your signal 2 which is provided by the T helper cell and signal 1 is by the own BCR, B cell receptor. Okay. Now we come to the next question. A B cell receives strong prolonged signal 1, that is BCR engagement, but no signal 2, T cell help. The most likely outcome is isotype switching to IgG without proliferation, immediate differentiation into memory T cell C. Yeah, good. So induction of energy, that is rendering the B cell functionally unresponsive. Yes, great. Right. Okay. Now we have another MCQ here. Which feature is common to both MHC class I and class II peptide binding groups despite their structural differences? Both are closed at the ends, strictly limiting peptide length. Both are formed from two membrane distal domains, one contributed by each polymorphic chain in class 2. Yeah, great. Yes, so B is the right answer. And why is A not the right answer? Because class 2 is not closed at the end and it is not limited by the peptide length. Okay. Okay. Now, a patient's cells show normal MHC-I surface expression but a complete absence of cross-presentation capacity in dendritic cells specifically, with conventional MHC-I presentation unaffected in other cell types. This is the most consistent with a defect specifically affecting. Correct. So the crosstalk between your type 1 and type 2 MHC is not happening properly, right? And that is why B is the right answer. Okay. Which statement about the conformational versus linear B cell epitope is correct? Conformational epitopes are always recognized by T cells. Linear epitopes remain recognized after denaturation since they depend only on the primary sequence. Yeah, good. So B is the right answer, right? So linear epitopes, they are the sequence, dependent upon the sequence and not the 3D structure. Okay, I think this is our last MCQ. Let's see. In the context of the MHC restricted T cell activation, which of the following pairs is incorrectly matched? Right. So C is incorrectly matched, right? All the other three are correctly matched. So thank you. That was all for today. These are your references once again. Okay. Thank you so much for paying attention in today's class. Okay. Acha, I'll give my mail ID once again, like I always do. If you have any mails, please send me. If I'm not able to respond, I'm sorry. I will respond to you. Okay, in case I forgot, I'm sorry. This is my mail ID. Just in case you need to convey anything, any feedback, any additional questions, or anything else, you can always mail me. Okay, this is my mail ID. Thank you so much to everyone. Yeah, sure. If you have anything to ask, you can ask me. Do you have anything to ask? Otherwise, we can end the call for everyone. Okay. Thank you so much. Thanks to you all. Okay. So let's end the call and we will meet again next week. But most probably I will change the date next week because I have one seminar. And so I'll have to present there. So every year we have around 2 presentations where we have to show the professors that we are doing work, and that allows us to get our stipend. Otherwise, okay, so then I have that next week. So we'll see. Maybe I will. Maybe we'll meet next week, or maybe we will meet on Tuesday. I'll tell you. Okay. NPTEL will send you that mail. Okay. Thank you so much. Bye bye. Have a great weekend. Oh no, sorry, this is not the weekend. Okay. Have a great week. Okay. See you.